In Topic 8 for AQA A-Level Biology, students need to understand the control of gene expression. One subtopic within this part of the specification covers the impacts of epigenetics on gene expression. Firstly, you need to understand some definitions such as the keywords epigenetics and epigenome. Epigenetics is the heritable changes in gene function due to environmental factors such as stress and toxins without changing the DNA base sequence. This is different to mutations which do change the DNA base sequence. The epigenome, however, is a layer of chemicals often referred to as tags which cover the DNA histone complex. The epigenome determines the shape of the DNA histone complex. There is strong evidence to suggest the influence of environmental factors on gene expression. Some of this evidence comes from studies on gestational diabetes. Gestational diabetes is a condition in which high blood sugar develops in the parent during pregnancy and usually disappears after giving birth. Some studies found that in humans, when the mum has this condition, the fetus is exposed to high concentrations of glucose, which can increase their likelihood of also developing gestational diabetes. Studies from nurtured rats show that the stress response genes are switched on in a rat pup which was raised by a relaxed and well-nurturing mum. Further evidence has been found from identical twins. Identical twins share 100% of their DNA. If one of these twins has schizophrenia, the likelihood of the other twin developing schizophrenia is 50%. This percentage is significantly lower in non-identical twins, which indicates that genes influence the likelihood of developing schizophrenia. But it also indicates that the chance of developing the condition is not only due to genes, but also due to environmental factors, as otherwise the percentage would have been 100. As well as understanding key definitions and evidence for epigenetics, you need to understand how environmental factors can impact the expression of a gene. Here you can see condensed DNA, also referred to as heterochromatin, and DNA which is not condensed, which is also referred to as euchromatin. In heterochromatin, the DNA is tightly packed around histones, reducing the likelihood of transcription factors and RNA polymerase from accessing the DNA. As a result, the gene that this DNA sequence codes for is less likely to be transcribed and therefore translated. This is the opposite for euchromatin. There are two types of reactions which can affect how condensed the DNA is, determine whether it will form a heterochromatin or a euchromatin structure. These reactions are acetylation, which adds acetyl groups, or methylation, which adds methyl groups to DNA. Acetylation decreases the positive charges on the histones, which reduces their attraction to phosphate groups on DNA. This causes a weak association between DNA and histones. As a result, the DNA is more likely to be transcribed and therefore translated. A way to remember this is to use the acronym MELAN as an abbreviation which stands for more acetylation, yes transcription, less acetylation, no transcription. In methylation, however, the addition of a methyl group prevents transcription by preventing transcription factors from binding to the DNA and by attracting proteins which condense the DNA histone complex.